Good morning. My name's Adam, and we're here today for your Discovery at Home Plant Biology Experimentation Program titled Ready, Set, Grow. In this program, you will investigate variables that affect plants' life cycles and growth. After hearing more about plants and their needs from Jill, you'll be better equipped to ask your own scientific questions and set up your experiment. Over the next few days, weeks, or months, you can continue to make observations to collect details to help form an answer. Those of you joining us today are in for a bittersweet treat, like Dad's old-fashioned licorice. Ready, Set, Grow on the last Thursday in June marks the 16th and final episode of Discovery at Home. I would like to thank all of you so much for your support over the last 2 months and invite you to join us at 11 after the break for the results of the Show Us Your Science contest. But I'm getting ahead of myself. We still have a program to run before I get to the thank yous, goodbyes, and celebrations, or the crying on camera. In a few minutes, Jill is going to take you through a bit of brightly colored background information that will hopefully introduce you to some new and interesting plants, as well as give you some different perspectives on the life cycles and abilities of some plants that you might already know. She'll go on to help you set up a fair and well-controlled scientific experiment. When I'm talking about control, I mean controlling variables, keeping everything the same except for the one thing that you want to test. If you change more than one thing at a time, you won't know what was responsible for the change in the result. In contrast, and while people are still joining and getting settled, I'll update you on my non-scientific back deck potato growing experiment. I say non-scientific because I did not try to properly control my variables. I just had way too many seed potatoes to start with. I didn't want them to go to waste, so I figured I'd just see what happens. I hope you'll see why my lack of control will really decrease my ability to draw meaningful conclusions to make decisions next year. But hey, I'll still end up with more potatoes this harvest. So the start of this potato story starts with something kind of like these. These were food potatoes that have been left around to start sprouting their eyes. But you see how these guys are kind of wrinkly? These weren't meant to be seed potatoes. They were stored for consumption and then left around a bit. So I started with nice firm looking potatoes and started to sprout them. So the way potatoes usually grow will be in the ground and start sending out these eyes. And they'll send out bits of stem under the ground that grow these tubers, which is the part that we eat. And then they also put big leafy parts into the air. When you grow them to try to maximize your amount of potatoes, you do this thing called hilling up. So if they're just growing in the ground, once you get some greenery, you add some dirt, and you keep adding dirt and dirt to make more of the plant be underground, and it will produce more tubers, more edible potatoes. So apparently some farmers will do those inside tires or bins or build it up with rock or brick. But you can also do a smaller scale in sort of any container, in a bag, and keep adding soil in your bag until it's topped up. So I'm going to show you some pictures from the last 2 months of my potatoes. Here's my first one, out to sprout on May the 2nd, so almost 2 full months ago now. And they took about 2 weeks to get some good sprouts on them before I planted them. Here they are almost a month after out to sprout, and you see just the first leaves are starting to poke out of the soil. So when I say a non-scientific test here, the bag was the purpose-built potato bag. And then with those extra potatoes, I was reading online, people say you can grow potatoes in anything. So I put them in an old cardboard box, which is starting to strain under the watering and weight now. But here's our side by side. Let's see if what I'm wondering is if the bag is really worth it in the first place. Here now is day 42. So this is just earlier in June, and you see them starting to get quite leafy. And the box is off to a strong start here. This here is 7 days after hilling up. So they had sprouted, and I added more soil to them, and they're growing greenery again. Now here's a much more recent one. They're getting pretty big. I put a meter stick here down to the base of the deck. So when they were planted, they had about 5 centimeters of soil in the very bottom. The seed potatoes were placed there, and then I added more soil to just cover the potatoes by about 1 centimeter. So the seed potatoes themselves are starting at about the same level and growing upwards. You'd see at this point last week, the box was up to about 50 centimeters and the bag was maybe 45 or so. And they started going really well. Here is yesterday, and the box is up above 60 centimeters, and the bag is still doing pretty well at about 50. But you see that really explosive growth out of the top of the box. So some of the variables that I didn't control well is that I selected what I thought, in my non-professional potato opinion, would be the best potatoes and put them in the bag and then put extra ones in the box. But maybe I didn't know what I was talking about when I was choosing what I thought were the strongest looking potatoes. And hilling them up with 4 in there was kind of awkward because I couldn't sort of add the soil too equally because I didn't want to completely cover over some. So the soil addition was not very well controlled. It would have been better to have multiple boxes and multiple bags and add it in a more controlled way. Here are some of my notes. And the turnaround. So I was a little suspicious that some of the smaller ones were in the back, and I saw a little bit of burning on the leaves from the heat coming off the siding of the house. So I spun them around yesterday to try to get the littler ones to the front, maybe give them more sun exposure or better temperature control. So my variables are all over the place, so I won't know at the end if the box was better than the bag. But like I said, I'll have a bunch of potatoes, hopefully. The last thing I'll show you with the potatoes is that I've been taking notes the whole time. So this way I know when things were done and added and some observations. So this is just a random Excel sheet that I was keeping track of. I didn't know I was going to get to use it this way, but I thought it could be useful to have some good notes. Well, buds, that's enough about the spuds. If you missed my intro, my name's Adam, and I'm one of the science educators at the Discovery Center. Welcome everyone to our final episode of Discovery at Home. Ready, set, grow. Today's program is all about investigating what plants need to live and thrive. Jill is just about to help you get well rooted in some plant biology and grow your own scientific question and experiment. Over the next few days, weeks, or months, you can continue to make observations to collect details to help answer your question. Even though our Show Us Your Science contest has ended, with the winners being announced by me in about an hour, I still very much hope you'll share with us what you've planted and observed on Flipgrid and our social media pages. But before I leave, let's run her over to Jill. You'll see why that's a plant pun joke, or at least an attempt at one, in a few minutes.
Hello and welcome to the Ready, Set, Grow workshop. My name is Jillian and I'm a science educator at the Discovery Center. Over the course of this workshop and the weeks of observation to follow, we will investigate what plants need to grow and be healthy so they can continue through their life cycle. We will learn all about the life cycle of a seed plant and some signs to help us determine if a plant is healthy or not. This workshop can be extended into a fun, summer-long project if you'd like to keep going with your plant observations, especially if you choose a plant that has a life cycle lasting over a few months, like a pepper or tomato plant. Let's start off with a definition of a plant to dig further into today's workshop. Plants can be defined as living things that have a life cycle, create oxygen, and respond to their environment. Humans have a long history of using plants for food, like my fruits and vegetables here, and using plants as medicine, like echinacea, to ease cold symptoms, and ginger root to help with nausea and motion sickness. We also use plants to make our surroundings beautiful, like flowers in our garden, and we can use plants to provide privacy and protection from the sun, like hedges around someone's yard, or a tree purposely planted for shade. Like humans, plants have certain needs to survive, but their needs are different from ours. As humans, we need to go find our food and eat it in order to have energy. Plants, however, are anchored into the ground by structures called roots, so they can't just wander to find their food. Instead, plants use their leaves, like tiny little solar panels, to soak in the sun's energy and use it to make their own food. Plants also need water and carbon dioxide from the air for this process. To help you better understand plant needs, we will observe parts of a plant's life cycle from a seed or sprout to later stages. Let me walk you through the stages of the life cycle for a seed plant so you know roughly what to expect. The life cycle of a seed plant, a maple tree for example, starts with new life growing in a seed. The seed has food stored on the inside and a tough seed coat on the outside to protect it as it travels long distances in the wind, through water, or by taking a ride on an animal's body, or even in their poop. Here are some examples of different seeds. These are green bean seeds, maple seeds, bell pepper seeds, and sunflower seeds. If the conditions in the soil are just right, the seed will start to grow by sprouting. This is called germination. Here, under our microscope, we can see some radish seeds germinating, and I also have an example of a maple seed germinating out in the wild. The seeds need warmth, water, and air to germinate. If the soil is too cold, too hot, too dry, or packed too tightly, and the seed can't get air, it will not germinate. Once a seed has sprouted, it will use the food stored inside the seed to help it grow. When the sprout's food is used up, it searches for more, sending out its first roots into the soil. The shoot will also grow upwards and poke out through the soil to reach the sunlight. The young plant becomes a seedling when it grows its first 2 leaves. Here's a seedling out in the woods behind my house. These leaves crack open the seed so the plant can reach the sun's energy. The seedling's leaves take in sunlight and its roots take up nutrients and water from the soil. The leaves continue to grow and its roots branch out to form a root system. If the seedling has enough nutrients available and can grow a sturdy root system, it will start to thicken its stem, grow taller, and get ready for more leaves in the next step. The next step in the life cycle is the vegetative stage. This is when a plant forms a sturdy stem or trunk for trees like our maple here. The stem grows branches, and the branches grow more leaves. The root system keeps branching and growing as well. The plant will continue to grow and branch in the vegetative stage until it is mature and ready to reproduce. Trees have a very long life cycle and sometimes spend more than half of a human lifetime in the vegetative stage, like my little tree here. In the reproductive stage, plants get ready to produce new life. For a seed plant, this starts with growing a flower that makes pollen. Did you know that maple trees also produce flowers? Let's have a look at one I snapped in my backyard. Here we see a nice red flower with lots of little pollen-producing parts getting ready to be pollinated. The plants get a little help from some flying insects for this part, like my bumblebee here. Bumblebees and worker honeybees collect pollen and nectar from flowers. The pollen gets stuck on the hairs on their body, and as the bees go about their day, they help to spread the pollen to other flowers so they can make seeds. For maple trees, seeds are produced 30 to 60 years into the tree's life cycle. Maple seeds have special wings to help them sail in the wind and reach different locations. Other seeds have different adaptations to catch a ride in the wind. Think of the fluffy part of a dandelion seed. They kind of remind me of little parachutes. It's important to recognize that seeds are not the only way that plants reproduce. Some plants use special horizontal or sideways growing stems above the ground called a stolon, or below the ground called a rhizome. These special stems send up new buds or shoots that will become a new plant. Other plants use tiny spores, which are spread much the same way as a seed, but are much smaller and do not have a seed coat to protect them. Some plants only reproduce one way, but others use more than one method to create new life. Strawberry plants can use seeds and stolons, and ferns can use rhizomes and spores. Once a plant can reproduce, the cycle is complete. But this doesn't always mean that the plant dies. Some plants, like trees and perennial flowers in the garden, will continue to live for many years and can create new life each year. Now that we've walked through the life cycle of a plant, it's almost time to start designing our experiment. But first, how can we tell if plants are healthy, meaning their needs are being met? Well, healthy plants will continue to have new growth and their leaves should be a bright, even green color. Unless the plant normally has spots or stripes, like something like this. Most common vegetable plants and outdoor flowering plants have solid green leaves. The leaves on a healthy plant should be firm, not droopy or crispy. Unhealthy plants may have leaves that are pale, turning yellow, or even turning brown and wrinkly, and the plant may have a tall and skinny stem with few leaves. Now we're all set up to talk about today's experiment. Our goal is to carry out an experiment to investigate the needs of plants. We will plant seeds or sprouts in different growing conditions and observe their growth over time to learn about their needs. Different growing conditions could be sun or darkness, giving plants water or no water, plants with soil or no soil, like rocks, or any other factor related to plant needs that you want to investigate. Since our activity focuses on what plants need in order to grow successfully, it doesn't matter if you choose a plant with a short life cycle, like a dandelion, or with a slightly longer life cycle, like a fruit or vegetable, because we will only need to track their growth and health over 1 month. It will be difficult to answer your question, however, if you choose a plant with a very long life cycle, like a tree or bush. This is because even when their needs are being met, they grow very slowly, so it will be difficult to notice obvious changes during our 1 month of observations. Let's get ready to dig in the dirt. Here are the materials you will need for today's activity: the observation chart from our learning guide or a piece of paper, a measuring cup for water, at least 2 identical containers, but I would recommend gathering 6 if you can, and I'll explain why later on. You'll also need something to poke holes if your container doesn't already have them. You'll need a permanent marker, and of course a growing medium like potting soil, compost, or peat moss. You'll also need several of the same seeds, sprouts, or mature plant parts to regrow, and make sure you have enough for each container. Without having to go out and buy seeds, you could harvest them from a fruit or vegetable you already have, like an apple or a bell pepper. Or you could go out and find seeds in nature. Dandelion seeds are one option that are easy to find in the summer. Seeds can take some time to get growing, so if you want to see results quicker, you could use something already sprouting, like a bean sprout, a potato with eyes, or a sprouting onion or garlic bulb. For even faster results, you could try to regrow parts of a mature plant like greenery from the tops of carrots, or the green parts at the bottom of green onions, or the bottom of a mature lettuce plant. Here are some optional supplies. You may also want a garden shovel to help you get digging, or garden gloves. You could also just dig with your hands. Depending on how you want to plan to gather your observations, you may also want a ruler and a ball of string and scissors handy. That's to help you measure your plant's growth. Now that we've gathered our supplies, let's design our experiment. Start by brainstorming some questions you have about plant needs and choose one question you'd like to investigate through an experiment. Here are some example questions to get you started. Can plants grow without sunlight? Can they survive without soil? Can plants grow without water? What type of water is best for growing plants, fresh water or salt water? You can use any of my example questions to design your experiment or come up with your own. The question I plan to investigate is, how does darkness affect the growth of a carrot top? I plan to observe a carrot top planted in a container on my bright windowsill and one planted in a container placed in a dark cabinet to see if the carrot in the darkness can grow. If it does, I want to see if its growth is different from the plant in the sunny windowsill. Next, use the observation chart or a piece of paper to help you keep track of your experiment setup. Write what you planted, that's the seed type, the sprout, or mature plant part you used, and write the date you planted it. Keep this chart nearby so you can keep track of any changes in your plants over the next few weeks. You could observe how much growth in centimeters you see each week and any changes in what they look like over time to help you determine their health. But we'll get back to observations in just a bit. If you haven't already, it's time to pick your containers for planting. It's best to use containers that have holes for water drainage. But if your containers don't have holes, you can use something to poke them. It's best to choose a material made of a soft plastic to do this. If you do need to poke holes in your containers, ask an adult for help because you will need a sharp tool to do this. I mentioned earlier to aim for 6 containers. This is because it's always a good idea to have more than one trial for each growing condition. In our case, that means more than one container for each growing condition in our experiment. Doing the same experiment a few times is called multiple trials. And in our experiment, this can be done all at once by having a few containers for each of the 2 growing conditions. Doing multiple trials is important so that we can make sure the results of our experiment were not due to a random factor. Imagine if you only had one container for each of your 2 conditions and only planted 1 seed in each, but neither of them sprouted. This certainly wouldn't help you answer your question. If you have several seeds, it's not likely that every seed wouldn't sprout, so you can see why it's important to have more than one. Since we have multiple containers, use a permanent marker to add a number or label to each container so you don't mix them up. For my conditions, I have sunlight and darkness. Make sure you write these labels on your observation chart too. So I labeled my containers here sunlight A, B, and C, or darkness A, B, and C. The next step is to fill your containers with growing medium, in my case soil, almost to the top, but leave a little bit of space to add some more later on. Now poke a shallow hole in each cup and place your seed, your sprout, or your mature plant part into the hole. If you're using seeds, I always recommend planting 2 to 3 seeds in each container in case some of them don't germinate. If they do all sprout and the plants get too squished later on, they can always be separated into different containers once you are done with your observations. Now cover your seeds with growing medium so that it is completely covered and there's at least a few centimeters of that growing medium on top. As you can see, I'm making a mess here, so it might be nice to do this outside or protect your work surface. Next, water your soil so that it is damp but not muddy. I made sure to use a measuring cup to make sure both containers receive the same amount of water. My containers are small, so I measured out 100 milliliters or almost half a cup for baking measurements. If your container is larger, you can always use more water to get the soil evenly damp. Just make sure you give the same amount to each container. Place your containers in a spot where they can stay for the next month as you make your observations. Once you have your containers in their final location, stop for a moment to assess that you have designed a fair experiment and make any changes if needed. A fair experiment means you have only changed one thing at a time. For example, with my question, how does darkness affect the growth of a carrot top, I will want to make sure both containers get the same conditions except the amount of light they receive. I use the same type and size of container, the same type and amount of growing medium or soil. I planted the same thing in each container, a carrot top, and will water them the same amount each time. I even kept them in the same room in my house so that the temperature and humidity were approximately the same. One is currently sitting on the windowsill above my kitchen sink where it gets bright light, and the other is in the cabinet below the sink in total darkness. Once you're satisfied that you have a fair experiment, it's time to be patient while your plants get ready to germinate. Remember that it could take a few days to a few weeks to see the first sprout, depending on what you planted. As soon as one plant sprouts out above your growing medium, write down how many days it took to sprout and write down some observations. What do you notice? What does the sprout look like? Gather observations each week as your plants grow to help answer your question. Good observations will relate to your plant's health and growth. Keep in mind the things that we described earlier to help us determine if a plant is healthy. Ask yourself, what do you notice? Has anything changed? How can you best describe what you noticed? For example, you could use words to describe the colors and textures of the leaves, or draw a picture of your plant each week and color them in. Are there any changes that could be measured or described using numbers? What you observe is up to you, but here are some suggestions. Measure how tall your plant is each week and track their growth. You can place a ruler so that the edge at that 0 centimeter mark is right against the dirt at the base of your plant. Hold your ruler straight up and check to see which of the longer centimeter lines the tallest part of your plant is closest to. If your plant goes past its closest centimeter line, count how many of these short lines it is past that long line. These short lines are millimeters. If my plant reached 3 of the short lines past the 4 centimeter mark, it is 4 centimeters plus 3 millimeters tall. Or to get more advanced with decimals, that's 4.3 centimeters. If you don't have a ruler and want to measure plant growth another way, you can use a string. Start by holding one end of the string at the surface of the dirt, pull the string straight up, and when it gets to the very tallest part of your plant, that's where you'll cut. Careful not to snip your plant. Now you can take this piece of string, glue it into your observation chart, and continue to do this each week to see how much your plants have grown. You can also observe color or texture differences in your plants. Is one greener, paler, wrinklier, or droopier than the other? And see if you notice when the plants transition to different stages in their life cycle, like from seed to sprout when they germinate, and then onto a vegetative plant. Gather observations and information from several sources so you have strong evidence to answer your question. Here are some other ways you can gather evidence. Observe the same type of plant growing in different conditions out in nature over a few weeks. Read a book or online article about your plant's growth and its requirements. Or speak with a longtime gardener or botanist, that's a plant scientist, if you know one. Use the observation chart to organize what you have observed during your experiment and any other information you've gathered to help answer your question. Once you've finished organizing, compare your results from plants in each of the 2 growing conditions. Look for similarities and differences that may help you answer your question. For example, do plants in one growing condition look healthier than those in the other? What evidence do you have for this? Did plants in one condition grow taller or produce more leaves? But here's an important point to consider. Does having a taller plant always mean that it's healthier? What other observations could help you determine if a tall plant is healthy, meaning its needs are being met, or if it's unhealthy, meaning they're not? Once you've finished comparing results and have answered your question, the last step is to share your results. Tell the Discovery Center, a friend, or family member about your experiment. Ask yourself, what have you learned about plants and what they need to survive? How can you use this information to help somebody else grow a plant? And why is understanding plant needs important to us humans? A great place to start is the Discovery Center's Ready, Set, Grow Flipgrid. I can't wait to see what you get growing and wish you all sunny skies and happy planting.
Thanks a lot, Jill. Viewers, if you've tuned in after 10 and missed my intro or some of Jill's information on making observations and controlling variables in plants' growth and life cycles, you can rewind and watch now or later. Doing so now might help you set up today's activity. You can also find the learning guide with suggested supplies on the Discovery at Home webpage. Before I get ready to send you off, I'd like to share a video of Jill's pet shamrock. This is really a bit of a bonus, but I'm going to try to frame it thinking about experimentation and observation. What if we ask the question, can plants move? More specifically, can shamrocks make themselves move? We'd have to control variables like airflow to make sure that a breeze isn't blowing it around. I have my pet shamrock here. Incidentally, Jill and I both have some. And so, if we took observations over a short period of time, we might conclude that the answer is no. Jill was able to set up a camera for a time-lapse video of her shamrock. I'll play it and ask you to make your own observations. It's 4 hours of video sped up to about 17 seconds. Here it is for you. Oh, a little slow. Perfect. Can you see any movement in the shamrock? I'll replay that once more for you while I'm talking about it a bit. When it's mostly in the shade, I can see it moving around a little bit slowly, kind of tweaking its leaves, maybe looking for more sun. And then we see when the sun shifts later in the day and it gets a bit of full sun, it says, no, that's too much, and it seems to settle down a little bit. Mine here has been fairly recently replanted, and the growth exploded after the replanting. I'll talk to you later about the importance of replanting plants in pots if you're trying to make them live for a long time. And it's been very funny watching mine because it reaches out towards the window and it's put all these flowers out recently. And it sort of goes to sleep either at night or if it gets too much sun. I was thinking I'm going to spin it around for you and see if you can guess which way was facing the window most recently. This guy had to take a car ride, and they're usually pretty picky, so I'm glad it looks so happy even after the ride. I'm not 100% sure myself, but I'm pretty confident that this was the window direction, where you can see the reaching towards. Well, now it's your turn to ask your scientific question and think about your experimental design. By that, I mean think about the variables, the things that can change, and figure out how to control them to focus on your question. In this sort of program, where you might be just setting up now and not have any observations yet, I want to make sure to encourage you to share anything that you think might be interesting, whether that's your scientific experimental setup, other observations of plant life and growth, or examples of interconnectedness of plants, humans, and our environment. I'll be able to watch for submissions on Flipgrid, Twitter, and Facebook to feature after the break until 5 minutes before we're back at 11. Now that our Show Us Your Science contest is over, I get to announce the winners, yes, that's winners with an S, of the Science at Home prize packs after the break. If you have questions, please reach out to us on the Discovery at Home page. Myself and our team of educators will be here to help you. Then, tune back in at 11 so we can see what experimental setups and plant observations and experiences others have shared. Welcome back, everyone. If you didn't see me before, I'm Adam from the Discovery Center. Today, we're investigating characteristics and needs of plants while they live and grow. We set up scientific experiments, identifying and controlling as many factors as possible to test a question. In a few minutes, I'll share some updates on Jill's carrot regrowth experiment, but first, let's take a look at our viewer submissions. Our first one here has a question. I'm going to try to put the question up and pause it for you. So the viewer's asking, do prayer plants grow better in water or soil? Let's have a look. We see our two samples. We have a rooted cutting in water. So the stems were cut, and this experimenter is trying to get it to make new roots. They ask, if I don't have more prayer plant cuttings, but I use another plant in water and in soil, is this doing multiple trials? Is my experiment still fair? I guess in that case, it depends what question you're asking, because it isn't multiple trials of do prayer plants do better in soil or water. You would need multiple cuttings of the same plant in order to test that. When we do 3 of something, we call it triplicate. So if you want to do multiple trials in triplicate, you would need 6 cuttings in total, 3 that you do in water and 3 that you do in the soil. The reason I said that bit of it depends what question you're asking, if you wanted to say, take a survey of different plants and have a bunch of different plants that each have 1 cutting in soil or water, you could think about that as multiple trials. But the really important part of that multiple trials is keeping everything the same and doing that in triplicate. Some scientists will say, if you don't know it in triplicate, you don't know it, because just one data point could be anything. You need a few to see if there's something funny about one thing. Like I was speculating about my potatoes, it isn't replicated enough. I don't know if that's just one bad potato or what. I would need more trials to draw better conclusions. Let's have a look at our next submitter. This one, they titled it Better Basil. Let's take a look. We see some detail notes here, starting almost a full month ago now, from some basil cuttings. Nice labeling, very important. Looks like it has a little rosemary neighbor. Oh, look at all those roots. That looks like a poor result. There we go. That bit of gravel in there is supposed to help drainage, so the plants don't get wet feet. That is what they call it if the roots are sitting in water. This is why Jill mentioned it's important to poke holes in the bottom. Same water source here for the two cuttings. So they're wondering if the cuttings will grow better in the can or in the communal box. And this is an important bit here that I don't think Jill did talk about it. How do you want to judge success? Whether you're going to do that by measuring or do it what we call qualitatively, looking at the qualities of the leaves, making sure everything looks healthy. This poster here said, how do you measure better, in quotes, growth? So it could be the number of new leaves, it could be the height of cuttings, and new increased height. So they're looking for a few different growth factors, which is, that's a nice way to do it. Instead of having just one thing, just saying taller is better, looking at different characteristics makes a lot of sense to me. That is it for our viewer submissions today, but I have a question somebody asked. They asked, effectively, are mushrooms plants? And this is a great one because it's based on sort of behavioral observations, right? Mushrooms seem to be sessile, they don't move, and they appear to grow like plants, kind of in the same environments. But kind of amazing to think of, we are more closely related to mushrooms, to fungi, than both of us are to plants. So, like us, mushrooms breathe oxygen. They don't do any photosynthesis, and they need to eat. So when you see them sitting there looking like they're growing like plants, they're most likely eating what's below them. They can also have some long, off the top of my head, I don't remember the word for it. It's kind of like roots, but they definitely aren't roots. So where you see the little fruiting body, the bit that we think of as the mushroom, they could have parts of their body going down into the soil or into the rotten log, and that's where they could be eating. And then what we see is a reproductive part that produces spores, like the fern that Jill showed you. So there's a lot of examples of sort of convergent evolution when two different groups come together in some characteristics because maybe they have a similar lifestyle or similar environmental conditions. Or you might argue that animals are mostly divergent evolution, that if the ancestor was sessile, which likely was, but I'd have to look that up a bit, then us moving around, we're the odd ones out, that we've gone off in a different direction and learned to move around much more quickly. So as you see, there is some plant movement. So I think that is it for my viewers now. Let's now have a look at Jill's carrots. I'm going to switch my presentation here to the other one with the carrots. Good. So Jill set up this bit where she ate the root or the tuber of the carrot, and then she had the top with the greenery cut off. So just that kind of like last little slice of carrot, and wanted to see how it would regrow. And she controlled her variables well, with her one test being light versus dark. They're in the same room, so hopefully very similar conditions. One gets sun, and one is hidden, I think, under the sink, she said. Let's have a look at some of her pictures. Here's 3 days after the initial planting when she first saw a sprout, only under the full sun. See, a normal little green-looking sprout, nothing too surprising. Now, at day 7, we start to see some growth that we can compare and make some observations with. So, on the full sunlight, we see more greenery and we see more leaves. The darkness kind of looks like a bean sprout to me, where it has that kind of white, pale yellow look. Let's have a look even a little 2 days later, after some more growth. that we have some really nice leaves by day 9 under the full sunlight, but we have this long kind of shoot that makes me think of the potato stems that's all pale. So it looks like if there's no sun on it, it's not bothering to express this chlorophyll, it's not bothering to put the effort into growing leaves, it's searching, it's trying to grow far enough, presumably to come out of the soil or the leaf litter or whatever it would be in its natural environment, to find some sun. And unfortunately, if we look a little further on, they start to droop. So the thing about trying to regrow these carrots is they don't regrow their big carrot bottom. So it's likely, I'm speculating a bit, but it seems likely to me that they weren't getting enough water or nutrients through their roots, so they had enough energy to do this bit of sprout, but both of them started to fade after 2 weeks. Let's see, here's Jill's observation chart. I'll show you that she was keeping track of these with her 2 different entries here, one for sunlight and one for darkness. She noted the number of days to sprout above the soil, and the one in the sun did much quicker, but because it's not done in triplicate, we don't know whether that's a real result that we can draw conclusions from. It's certainly what we might expect to find, but we can't be confident without at least the triplicate. And she follows her observations all through and has made it fairly detailed, contrasting the different growth between the 2 plants. I have a couple more examples here to show you, not off the laptop, but next to me here. Another Jill example is these 2 mint plants. Oh, need 2 hands on this guy. Oh. So, to look over this one. So the story with these 2 plants are Jill had found a bit of mint growing in her garden. Mint is something that we use for food. It smells and tastes very nice, but it is an invasive plant. I'm pretty sure it's European, where it grows wild. So here it's not a local plant, and it sends out those runners. I think the subsurface, the rhizomes that Jill told you about. So they can sneak out of garden boxes and spread quite quickly and take over a garden. And they could even go underneath a fence and take over a neighbor's garden. So I do have some mint of my own at home, but I do keep them in pots so they don't take over. So these fairly aggressive plants, these guys started with the same cuttings on the same day, no, sorry, not cuttings, dug out of the same garden on the same day, planted in the same soil, and put in 2 different pots. The big guy is from Jill's, and the little one is from her mom's. She gave her mom a mint. And she told me that they were in pretty much the same sun conditions. It was full sun for part of the day. So it looks like the most significant uncontrolled variable was pot size, which shouldn't be too surprising here, that we got much more growth when there was more soil. There's more room for the roots to spread, and we got a much bigger plant in the big pot and a littler plant in the little pot. I mentioned something earlier about talking to you about repotting houseplants. And I'm going to bring my shamrock back in for a sec. I might have to clean up a bit. This guy is huge. Here we go. So, see how much bushier my shamrock is than the one that we showed from Jill. Mine looked a lot like Jill's about a month ago, and I hadn't repotted it for about 2 or 3 years. And I repotted it even in the same pot. So it didn't even get more soil, but it got new fluffy soil. The thing about pot soil is there's a lot of organics and it needs to hold on to water. But there aren't things like worms traveling through the soil to keep it mixed up a little bit. So it tends to compact over time. So most potted plants do better if you replant them every 1 to 3 years or so. So this is about a month after replanting in a bit of fresh soil in the same pot, and the growth exploded. It did tons of vegetative growth and now lots of reproductive growth with all of these flowers. So there's a lot of variables and a lot of things to think about with these potted plants if you're going to do longer and longer term things. Now, the last thing I'm going to show you with the plants here isn't really a controlled experiment. It's just showing another variable. These are two aloe vera plants. This one's Jill's and this one's mine. Jill's is looking a lot better, isn't it? So again, they haven't been controlled at all, but what I want to show you is look how healthy the leaves or whatever you call it on an aloe vera look in Jill's. We have nice even color, we have a lot of growth. Mine was a bit rescued. It was kind of sick before, it was left out, and we did a little bit of surgery, cut off some leaves or whatever there, and kind of stunted them there. There's a bit of new growth, but we made a mistake earlier in the spring. It was inside, not getting too much light, and we figured it's a cactus, it's good with the sun, so we'll put it out. And I should have known better because I know plants can get sunburns. My dad would always start plants in the spring in the greenhouse, and then when it came time to put them outside, it kind of made me think of like putting dogs out in the yard or something. He'd put them out only for a few hours during the day and cover some of the sensitive ones over with screen, and he'd acclimatize them to the full sun to get them out for the summer. I should have been more careful with the aloe, but again, thinking it's a cactus, it'll be fine in the sun, but you see this discoloration? That is sunburns on it. So the plants can get sunburns too. Many of them that can tolerate full sun adapt much better than humans do, but it takes time. I shouldn't have gone full sun on my aloe. Well, folks, I think Ready, Set, Grow has gone to seed. Usually, that expression means that something has become older and shabbier, kind of run down, referencing a plant that has exited the vegetative growth stage and spent a lot of its energy in the reproductive stage to produce seeds and be ready for the next year or next generation. Here, I don't mean to invoke the shabbiness part. I'm imagining this program as the vegetative stage, growing your knowledge. I hope we were able to plant a seed of curiosity and interest in careful observation and experimentation, and that you'll carry on with your experiments and try new things over the following days, weeks, or even months. Now that Ready, Set, Grow is wrapped up, it's time to turn my attention back to the end of Discovery at Home and announce the Show Us Your Science, Science at Home prize pack winners. Our winners are, I was thinking I should have got a drum for this. First one is Liam Hachet. Our second is Josiah S., then Nicholas B., Casey McGinnis, and Jesse Sellers. You can read them here. If I've mispronounced any of those, sorry. Since we don't necessarily have your contact information from your submissions, please email info@thediscoverycenter.ca. That's info@thediscoverycenter, Canadian RE.ca. If you're curious, and I tend to be about this sort of thing, of the 5 winners, 4 were from Discovery at Home submissions, 1 was from Bite Size Science. 2 came in on Flipgrid, 2 on Twitter, and 1 on Facebook. Congratulations to our draw winners, and congratulations to all of our submitters and viewers. I'm really genuinely happy that we were able to support your learning and having fun with science during this time. Thank you all so much for watching, doing, and sharing. I want to extend a particular thank you to all of our submitters. You made my job easier, better, and more interesting in the segments after the break. And I'm sure seeing your contributions and the questions and observations they generated helped your fellow viewers to learn and gain inspiration. I want to extend the same thanks and well wishes for my fellow science educators. You saw Jill, the anatomist, and myself, the chemist, played Jack and Jill of all trades, running various programs and acting as hosts for Discovery at Home. You also got to meet Jen, the one-time field correspondent and constant developer, without whom Jill and I wouldn't have had time to focus on hosting. Similarly, Sabrina, Natalie, Emily, and Jepna took time away from background and other work to share their expertise with you. Sabrina got to talk to you about rocks, which is where you find dinosaurs. Natalie got to share her academic and personal love of food with you. With Emily and Claudia, you got to explore some of the wonderful diversity of life on our planet. And Mission Specialist Jepna took you beyond our atmosphere to show you that the sky isn't the limit of human exploration or your imagination. Behind the scenes, our managers worked harder than I think we realized to make this all possible. People put on many hats. Jamie helped in program development, and our exhibits team, like Will and Chris, completely built and ran our studio in what is usually the third floor visitor lounge. They've been half-jokingly counting Will's hats, which means the number of different jobs that he's doing. I think I heard they're up to 33, with his helping me with my French for a project over the summer. I think that brings me to looking towards the future. We're busy here getting ready for opening up in July, so I hope to see some of you in person if you can visit the Discovery Center. And I hope you'll see me in some digital form in the future. From everyone here at the Discovery Center, thank you all. Stay curious this summer and beyond.